Now let's talk about meiosis, which is a series of steps that happen when a diploid cell divides and gives you 4 haploid cells. It's the series of divisions that happens that gives us sperm and egg. Now, before I go through in detail into steps of meiosis, I just wanted to remind you of mitosis. Remember, if you look in the lower panel, before mitosis could happen, there was an interphase preparatory step in which the cell became ready for cell division, and part of that readiness was to duplicate the chromosomes. So if you look in here, you have 2 pairs of chromosomes, and they are basically 1 chromatin on each, but then by the time interphase is over, each of these chromatins have a sister chromatin. So these are sister chromatids now, we call them. Okay? So then what happened? We went through the prophase and prometaphase stage. The chromosomes all lined up in the middle of the metaphase, on the metaphase plate, which is the middle of the cell. Sister chromatids were separated and then divided into 2 cells. That's mitosis. And there was 1 cell division. Meiosis is different because in meiosis, even though there is still this interphase preparatory steps where this cell gets ready for division and it duplicates its chromosomes, then right after this interphase, the steps are quite different. First of all, there are 2 consecutive cell divisions. You get cell division 1, which we call meiosis 1, and then cell division 2, which is meiosis 2. After the first cell division, we get 2 cells, and then 2 of each cells are going to give you 2 more cells. So at the end of meiosis, you start with 1 cell, which is diploid, and you get 4 cells at the end, which are haploid. With mitosis, we said mitosis is asexual reproduction, making more of the same. In this case, we're starting with a diploid. I also want to mention that haploid cells can also undergo mitosis. So you go through steps of mitosis and you get 2 cells that are identical to each other and to this cell with which we start with. So now let's look more in detail as to steps of meiosis. So the first step after the interphase is basically meiosis 1. The first stage is meiosis 1, and meiosis 1 we're going to only talk about these 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. So if you remember, with mitosis, we have prophase, and really the significance of prophase and prometaphase is that chromosomes kind of line each other up randomly, and then during this process they connect themselves to the spindle fibers through their kinetochore. And that's pretty much, and then another thing that happens is the nuclear membrane dissolves. Okay? In prophase 1, things are different in some ways. Some things are similar. So just like mitosis, these centrioles are going to duplicate. They move to the 2 ends of the cell. Nuclear membrane breaks down. And by the time you see prophase 1, chromosomes have already duplicated and they're condensed. But what you notice is that chromosomes are paired up. And not just any chromosome pairs up, it's actually homologous chromosomes pair up. So in this example that we're seeing, we have 3 pairs of chromosomes. So this cell starts with a total of 6 chromosomes, which are 3 pairs. So homologous chromosomes come together during prophase 1. They not only come together, they actually exchange genetic material. So if we had this blue chromosome here, this blue chromosome is going to swap some of its chromosome with the red one. So then this chromosome is going to be blue, 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 red. And it's going to be red, 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 blue. So there is genetic exchange happens. And the terminology for the structure of homologous chromosomes sitting next to one another and attaching to one another and exchanging parts is called a tetrad. Okay, so lots happens during prophase 1 in meiosis 1. Not only the nuclear membrane breaks down and then the centrosomes move towards the two ends of the cell and they start connecting to the centromeres of the chromosomes, but one major thing that happens is that homologous chromosomes actually pair up and exchange genetic material. Next, we go to metaphase 1, which kind of conceptually similar to metaphase of mitosis, except in mitosis, let's go back, all of the chromosomes line up and form a single line. There is no pairwise interaction. But when you look at metaphase 1, you see that these chromosomes that held hands in here, they continue to hold hands. So chromosomes line up on the metaphase plate during metaphase 1 pairwise. And you see that by this point, they're also connected to these fibers, these microfibers, right? Okay. So anaphase 1, which in mitosis separated sister chromatids, in anaphase 1, you don't separate sister chromatids. You see these sister chromatids are still attached. What you do in meiosis 1, you actually separate homologous chromosomes, such that by the time telophase and kinesis happens, on one end of the cell, you have one set of homologous chromosomes. In the other end of the cell, you get the other set of homologous chromosomes. So each end of the cell gets one of these pairs. So by the time meiosis 1 is finished, you're going to get two cells that are haploid. You start with a cell that is diploid, and you get two cells that are haploid. Now, cells that are not diploid or don't have an even number of homologous pairs, they cannot undergo meiosis, because meiosis depends on homologous chromosomes pairing up during prophase 1 and also lining up together during metaphase 1. So that was meiosis 1. After these two cells separate, then there is no more interphase. Okay, so then they go straight into prophase 2, and steps in meiosis 2 are pretty similar to what happens in mitosis. So again, these spindle bodies move towards the two ends of the cell, nuclear membrane breaks down, the chromosomes attach themselves to these spindle bodies, and then during metaphase 2, all the chromosomes form a single line. Okay, no more pairwise because there are no pairs. Then, just like mitosis, during anaphase 2, sister chromatids separate, and then you're going to get two cells from this top cell, two cells from this bottom cell. And since during prophase 1 there was exchange of genetic material, you see we got these two ends are not identical. These sister chromatids are no longer identical. So therefore this division doesn't give you identical cells. So by the time meiosis 2 is finished, you get 4 cells that are different from one another and also from the parent cell. So next we're going to talk about how meiosis and sexual reproduction leads to variation in sexually reproducing populations. Why is it that within a family, no matter how many children are produced, they are neither looking like each other or the parents?